Hi, Miss Velez. Yep. This is my recovery assignment for AP Bio, where I talk about topics. So I'm gonna do it in the order of the unit. So Unit 1, Unit 2, Unit 3. I have a checklist here of everything, but there's like nothing specific. And yeah, I'll just do everything I can to tell you what goes on in AP Bio. If there's anything I don't remember, it's probably yep. It's probably Unit 8, to be honest. So yeah, here we go. For starters, I'm gonna talk about Unit 1 and water first. So water is give me a second. Oxygen and two hydrogens bonded together by a covalent bond. Right? Oxygen is negative, the hydrogens are positive. So this causes water to be polar. Because water is polar, it also causes water to be a universal solvent. And that's a property of water. In other words, it can dissolve a lot of stuff. Water creates bonds with other water through hydrogen bonds. So the bonding of water molecules is cohesion. So yeah, water molecules bonding through hydrogen bond, or while adhesion is basically water bonding to other other surfaces. So yeah, this right here will be adhesion. Together, they create capillary action. Oh, other properties of water is density, which was a question surprisingly on the exam. So basically, water, right? Let's just pretend this is water. When it freezes to ice, it actually becomes less dense. And as a result, ice floats on top of water. And this is very helpful for the environment because it keeps animal life away from the elements. So it preserves whatever is below the ice. High specific heat, right? That's essentially water does not change temperature easily. So water will consistently remain a certain temperature, and it absorbs heat and releases heat at the same time. It's very useful for regulating climate and stuff, and it allows organisms to be very happy and um sorry, it allows organisms to still survive when it's in like winter time and not freeze, and in summertime and not cook to death. Another is heat of evaporation. This is very chaotic. Vaporization, which is when sweat cools, vaporizes, which cools the body temperature. There's surface tension, which is caused by cohesion, and basically it like creates a barrier on top of water. It allows water striders to walk on water. Okay, moving on. We're gonna talk about macromolecule that word. Give me a second. I have my pen too big. So First, we're going to talk about carbohydrates. So, its elements are carbon, hydrogen, oxygen in a 1-to-1 ratio. Uh, they often look like rings, carbon rings. The monomer of carbohydrates is monosaccharides. Yeah. The polymer is polysaccharides. Shocker. Shocking right there. Its function is for short term energy and structure. So, I guess examples of this would be glucose and starch for short term energy. That's not how you spell it at all. While for structure, it will be chitin and cellulose. So, yep, there's that. Now we're moving on to lipids. In other words, the group where they put everything in because it doesn't fit anything else. Elements is also carbon, hydrogen, and oxygen. However, it has a lot of carbon, hydrogen, while very little oxygen. So there's two different types of lipids. There can be saturated lipids and unsaturated. So basically, saturated lipids are long chains of carbon that they look very straight. Um, they don't have any breakage, while unsaturated does have breakage in it. These this causes saturated to become solid at room temperature and unsaturated to be liquidy during in room temperature. Monomer is fatty acids. Polymer is fats, waxes, oils. The function of these is to insulate and long-term energy. Well, to create the cell membrane. Examples of lipids would be steroids, phospholipids, which help create the cell membrane. I'm gonna just draw it because it's a long word and I don't feel like writing it right now. Oh yeah, oil, fat, wax. There was another I think mentioned glucagon or something. I think it was glucagon. If it's not, it's not. Okay, moving on. So we're going on to proteins. I don't think I'll have enough space to talk about proteins. But basically, elements. I'm just not gonna write the subgroups. Okay. So elements: hydrogen, carbon, oxygen, and nitrogen with sometimes sulfur. It looks something like this. So I forget what's here. I think it's a it's a carbon right here. So over here we have an amine group of. Over here we have a carboxyl, and down here we have the R group, the rest. And basically, this is the structure of a protein. You know, the monomer is amino acid. The polymer is polypeptide. Function, there's proteins have too many functions for its own good. So it can catalyze reactions, which are enzymes, communicate with cell. So that's your hormones and receptors. It can antibody, so fight for your immune system. There's the foldings for a protein. So we have 4 folding types. The first stage is just the polypeptide chain. The second stage, you'll get spirals, or I know it doesn't look well. So yeah, this is your alpha spirals and this is your beta sheets. These are done with weak bonds. This is done. Amino acid is done with peptide bonds. I think this one is also done with weak. It's done with weak bonds, so like hydrogen bonding. Tertiary is basically a structured protein, which is also done with weak bonds, but this is done through they're bonded based off of their R group, which is why the R group is very important. Weak bonds like disulfide bonds, hydrogen bonds, London dispersion bridge reaction. There's three types of bond. There's polar, which goes with polar. There is non-polar, which goes with non-polar, and there's the charged ones. So there's negative that goes to positive and positive that goes to negative. Non-polar, when it folds, will go on the inside because it can't. It's hydrophobic. Oh, now I remember something. Remember something. Hold on. Hydrophobic. Yeah, lipids are not water loving. Phospholipids, on the other hand, they have a hydrophilic head, so it's water loving, and two hydrophilic tails, making it, and not hydrophilic, hydrophobic tails, which means it doesn't like water. So okay, yeah, I finally remembered. Yeah, non-polar non-polar amino acids go on the inside because it cannot interact with water, while polar amino acids go on the outside when it folds. And then charged, I'm pretty certain it goes on the outside as well. Quaternary is basically combining two proteins, two or more proteins, to create one protein. This is also done with weak bonds. Is there anything else? Well, there's the types. You know, there's, like I said, I have here enzymes, hormones, receptors, antibodies. I'll just quickly talk about enzymes. I'm pretty sure I had that somewhere at some point, but I don't for some reason. Okay, enzymes. They catalyze reactions. Okay, they make reactions move faster. I guess that goes under number 3, unit 3. But we'll just talk about it right now. When an enzyme falls out of place, so either temperature or pH falls out of its optimal range, so where it best functions, the enzyme can become denatured, basically, meaning its substrate does not work anymore. For temperature, it goes up the higher the temperature until a certain point where it just stops working. While pH, all enzymes have their optimal pH. For our example, we're gonna say the optimal pH is right here, and it's a lot more acidic. So based off acidity, the more it gets to its optimal range, the much more it creates. Well, it functions, but once it goes past that, there's less energy involved. Denaturing is not good. This is more or less the general concept. What else? Oh yeah, enzymes are like designed as a lock and key, so they're meant to fit perfectly with your substrate. So this substrate right here, it would fit perfectly in the enzyme. This is called lock and key. When altered, it doesn't fit to the enzyme anymore. Yeah, I'll talk more about it when I get there. I'll talk about it in cell signaling. Finally, nucleic acids. Yay! So its elements are carbon, oxygen, hydrogen, nitrogen, and phosphorus. It's nucleotide. It's a monomer. It's a nucleotide. Nucleotide. Its polymer is, well, nucleic acid. Shocker. It functions to store genetic information as well as, I guess, energy, technically, since ATP is energy. Codes for proteins. Okay. Examples of this will be DNA, all RNA types, and ATP, technically. So the structure of a nucleotide, which creates amino acids, not amino acids, which creates nucleic acid, will be something like this. So we have our phosphate group right here. Then we have our carbon ring. So this is where the first three come in. And then we have our nitrogenous base right here. So phosphate, carbon ring, nitrogenous base. In Unit 6, I'll talk about DNA and RNA more in detail, because that's where we're going to talk about more of it. But yeah. Oh, I guess this is necessary. So there's, because I'm not going to talk about purines and pyramines. The purines. I might have this backwards, so keep that in mind. I always get it backwards at some point. Pyramines? Whatever. Purines will be adenine and guanine. These are the nitrogen bases. While pyramines, I don't know how to say it, will be cytosine, thymine, and uracil. So both of these codes have a hydrogen bond with adenine. So it really just depends on what type. So DNA would have pyramine, while RNA would have uracil, and guanine will always bond to cytosine. There is 2 hydrogen bonds between adenine and thymine or uracil, and there's 3 between guanine and cytosine. And that's all I have. No, not all. Fun thing is creating polymers and monomers, and with this we have the condensation reaction. So for this, I'll just be using carbon. So we have 2 carbons, but we want to have a polymer. So what needs to happen is that we need to remove water from here and here. So we'll remove 1 oxygen, 1 hydrogen, and then from here we'll remove 1 hydrogen. So by removing this, we get water, and this results in our polymer, a disaccharide, and a drop of water. And then here we have hydrolysis. Hydro, water, lysis, break. If this is creating a monomer, so we take that lovely carbon, we add water to it. Okay, we add it to here, and we result in 2 polymers again. Not 2 monomers. Jeez, I keep getting it confused. And yeah, that's the end of Unit 1 for me. Unit 2. So first we have organelles. This is very simple. I'll just be listing organelles and what they do. So we first have our nucleus, nucleus, however you say it. You know, it controls the cell. It stores DNA and technically RNA. It's where RNA is produced. Then we have the ERs. So rough, it has ribosomes on it, and it transports proteins to the Golgi. The smooth ER, it detoxifies and it produces lipids. Going from there, Golgi. So it modifies, it packages, it does produce carbohydrate. Yeah, but it modifies, packages lipids using vesicles. So, not lipids, just anything that it receives. So it can go around the cell or out the cell. There's lysosomes. Basically, it digests. It has a cell membrane, which has a lower pH. It also does apoptosis. In other words, it programs cell death. It digests waste or energy, I guess. It also recycles dead cell organelles to be used again later. We have our ribosome, which is the site of protein synthesis. There's vacuoles, which basically it stores food, waste, whatever. It stores it for later use to be destroyed later, to take it out the cell later. The large central vacuole is only in plants, and it not only stores specifically water, but it also creates turgor energy in the cell to make it much more stable for plants. Cell wall, which is only in plants, and it protects it, gives it structure. Cell membrane, it it controls what goes in and out. Chloroplast, it produces it's photosynthesis. It produces energy for plants. It's a plant specific. Mitochondria, it's the site of cellular respiration. And the final thing is cytoplasm, at least from what I remember, which just is like a jelly substance that holds everything together. It holds all the organelles in the cell membrane in the cell. Also, cytoplasm has the site of glycolysis, whatever you call it. Prokaryotes. So very simple. Eukaryotes are complex. All this has no double membrane, so the organelles do not have a double membrane. While here, in eukaryotes, we do have a double membrane. They're smaller. Eukaryotes tend to be larger. DNA is circular. Eukaryotes DNA is straight. There is no nucleus in prokaryotes. That's more or less the differences between the two. Moving on. The cell membrane. So, the cell membrane controls what goes in and out of the cell using phospholipids to create the bilayer. So, this is the mosaic fluid model. So, within it, we have a lot of stuff in here. We have this is cholesterol. So, cholesterol, it does stability. It prevents the cell membrane from being too smooth, too liquidy, I guess. It keeps stability within the cell wall. From there, we have transport protein. So basically, they allow things that doesn't naturally flow through to the enter the cell, or it could exit the cell. This is a glycoprotein with carbohydrates on top of it. So the carbs are there for cell signaling. Yeah, and that's basically so the reason it is structured the way it is because the phospholipid heads are, again, they're hydrophilic, while the hydrophobic tails are, well, hydrophobic. So they have the polar hydrophilic side outside, so it can talk with water and control what goes in and out. So that's all I have for cell membrane. Next we're gonna talk about transports types. So we have passive transport. This is everything that goes along the concentration gradient. So we're going from high to low. This does not require energy, no energy. So moving along, different types of passive transport. There's diffusion, which is just letting things go, small little nonpolar things go through the cell. So this will be like carbon dioxide. There's facilitated diffusion, which is using a transport protein to let things in and out of the cell. As long as it is a long gradient. There's osmosis, which is just water going through in and out of the cell. So yeah, now we're gonna move on to active transport. So unlike passive transport, it goes against the concentration gradient. It's basically like everything backwards for passive transport. So basically, it goes from low to high. It requires energy to actually occur, and the energy is mainly ATP. Different types. There's endocytosis and exocytosis. There's others, but they basically fall under these two categories, to be honest. It either enters the cell or exits the cell. An example of this would be the ion pumps, where you are taking out sodium and you bring in potassium. We're going to move on to osmolarity and water potential. So basically, water potential is how much water a cell can absorb. So this is where you get your tonics. So you get your hypertonic, your hypotonic, and your isotonic. When in a hypertonic solution, the cell will end up shriveling since water is going out of the cell. In a hypotonic solution, water expands the cell because water is going into the cell. And in a isotonic, water will be going in and out. Other stuff to know is that water moves from high water potential to low water potential. There's the equations to find a water potential. This is the equation to find the actual potential. I don't remember the second equation. Well, I'm not supposed to memorize these stuff. There's another equation that helps you find the water moves from high to low. So high water potential to low water potential. You go from high amounts of water to low amounts of water. It's simple diffusion. Yeah, I think that's really all I have to talk about for the rest of Unit 2. So Unit 3, we'll get into energy and energy stuff. So the laws of thermodynamics. Essentially, the one that I think we're supposed to remember is the one where you can't create energy, so energy cannot be created, only, I guess, recycled. It's because you reuse the same amount of energy again and again, one way or another. The other thing is energy is lost as heat. So there's free energy. That's what the symbol is, right? Gaining energy and losing energy. So when you gain energy, right, you digest it, you have energy to be used later. While lose energy, you're doing something. I do remember the ATP and ADP cycle. Basically, ATP, right? It has 3 phosphates. So when it's being used, so we're losing energy here. So for example, we're gonna use it for opening ion pump, right? We're opening this ion pump. We end up with ADP and 1 phosphate. We're done with the ion pump. So we do ADP goes through and becomes ATP again, making us gain energy. And basically, it's a cycle that goes on and on and on. And this is how ATP is recycled for the most part. You go again and again. I mean, it's not infinite. I don't think it is, but okay, moving on to photosynthesis. We love photosynthesis. So yeah, this is use light to make energy. The equation for this is carbon dioxide plus water plus light yields glucose and oxygen. So this occurs in the chloroplast. So there's two reactions that occur in the chloroplast. So we have our light dependent and we have our light independent, or in other words, the Calvin cycle. Now, I can tell you right now that I don't really remember much about this. Like the specifics to it, I can't really tell you much of the specifics because there's like particular numbers and stuff to it. But I can tell you the general idea. So here we have this one occurs inside the D thing, which is called the thylakoid, or whatever, and then the light independent occurs in the stroma, which is basically the cytoplasm of the chloroplast. Yeah. So in light dependent, we are producing ATP, we are producing NADPH and oxygen. First, we have to separate the water into proton and electrons. Yeah. So we can have our electron transfer chain. So yeah, light excites the electron at this point. So the electron will go enter the first photosystem, which is photosystem 3, where light continues to be absorbed by chlorophyll. The chlorophyll continues exciting the electron. So the electron will continue through the ETC, and then here we're pumping in protons using the electron that's passing through with chemiosmosis. Then it goes back here where it goes into photosystem 1, where we start producing we start exciting, I think, NADP plus, and then as a result the electron excites it and gives it and reduces the DP plus into NADPH. There's also a ATP synthase. Since we're pumping in hydrogen or proton, we have to eventually pump them out. So we send the hydrogen outside, and this causes ADP and the phosphate to become ATP. Yay! So that's how we end up producing, and obviously the remaining stuff, because we get the protons from water, so the oxygen will remain, and that's how we get oxygen. So the light independent occurs again in the stroma of the thylakoid. This is what produces NADP plus ADP plus 1 phosphate, and it produces glucose. So it's a cycle. I don't really remember this very well, So keep that in mind. But from what I remember, we take in the carbon dioxide. This also releases oxygen. So we take in carbon dioxide. Okay, so the carbon gets separated from the oxygen. So the oxygen kind of just gets tossed away. Well, that and or B gets used for glucose. But basically, the idea is the same. Carbon separates. We get these carbons, which connects to two other carbons, and create this one carbon. I think it was called like Rubin Vade. Well, we still need to produce glucose though. Some carbon gets left behind and continues. It gets returned into Rubin Vade or whatever it's called. Carbon dioxide gets introduced, and yeah, it's a cycle that goes on and on. It takes 2 cycles to create one one glucose, I believe. Moving on, we have cellular cellular respiration, which produces ATP. So we are producing ATP. It occurs in the mitochondria. Yay! Which is also double membrane, but I'm too lazy to draw another membrane. The equation for this is glucose plus yield, ATP, carbon dioxide, and water. I meant glucose and oxygen. So there's 3 steps. So first we'll talk about glycolysis or glycolysis. I really don't know what this actually is, which is breaking down glucose. It actually occurs in the cytoplasm before anything else. And basically, there is 2 ways it could go. This is anaerobic, so no oxygen is needed. So there's 2 ways this could go. We break down glucose, we go through everything, and then we go through fermentation, which could end up with either ethanol or lactic acid. And we're also getting NADH plus ATP. However, we also have to use ATP to have the process, so there's that. But if we do have oxygen, this is done with no oxygen. So no oxygen, right? But what if we do have oxygen? Well, we'll go into the mitochondria and we'll begin our second thingamajig. I'm sorry. Which is the Krebs cycle. Basically, glycolysis. It breaks down the glucose, and we get pyruvate. And that pyruvate gets turned into hexacola or something like that. And that is used in the Krebs cycle to produce more FADH. We produce some ATP. We produce carbon dioxide. And we produce NADH. Right? We're reducing all of these into the formats. From there, we go to our third cycle, which is the ETC, where we're actually producing. Oh, this one occurs. This. Let me put this in color so it's like easy to see. So right here, this occurs here in the middle. The ETC occurs on the crestae, or the inner membrane, whichever one you want to call it. The crestae are the folds within mitochondria. So basically, we are once again exciting electrons to pump in hydrogen or protons. So we start here. So electron goes through. It gets excited. I don't really remember what exactly it gets excited by. Meanwhile, hydrogen is being pumped through. I think all of these pump up. Eventually, we reach the ATP synthase, which converts ADP and phosphate into ATP. The hydrogen will bond to the oxygen, and this is how we produce our water. We produce NAD plus, I believe, ATP, and NAD, also water. The final thing I want to talk about in the symbiosis theory. So in this theory, basically a bigger prokaryote saw both chloroplast and mitochondria, and it absorbed them. They would get a home, so these two will get a home, while this will have will have organelles produce. It absorbed a nucleotide, not a nucleus. It absorbed ribosomes were like naturally in this. It absorbed a vacuole lysosome that absorbed the ER was, and basically the theory goes as follows. All eukaryotes come from prokaryotes. Prokaryotes got smaller prokaryotes that got absorbed by large prokaryotes, and eventually we get our eukaryotic cells from that. Which would explain why mitochondria and chloroplasts would have their own sort of energy to them. So evidence for this theory would be double membrane. It would be the DNA within it. It will have small little plasmids inside. And it's very similar to other. It's very similar to other prokaryotes out there. There is more. I know there is, but I just don't remember at the moment. And yeah, that's the end of Unit 3. Okay, we're moving on to Unit 4, which is cell signaling. So, we're starting off with, I guess, the 3 steps: reception, transduction, and response. So, reception is when a ligand connects to a receptor. The ligand is the signaling molecule. It kind of works like enzymes. So a specific ligand will bond to a specific receptor. This response ends up with transduction, which causes a cascade to occur where reactions keep occurring and occurring and occurring. Specifically for my example, because this is what I remember, we'll have a phosphorylation cascade where phosphate keeps being added and then subtracted to then remove to activate and deactivate the response as it goes down into response, which is just the outcome that the cell wants to receive after everything that happens. So the cell gets its response and releases it to be used. There's two types. There is there's protein signaling, which is using proteins like this to communicate a response. There's lipid signaling, so which uses lipids, so like estrogen, testosterone, to respond to get responses, right? The response can either occur in the nucleus or the cytoplasm, and different types of response. We have endocrine, which is within the cell, so we're sending one response to another part of the cell. Paracrine, which, if I'm not mistaken, it's the one where it's a short distance from each other. Juxtacrine is like literally right next to each other, and there's one where it's long distance, but I don't remember the name. Whatever it was, there's one that uses like the the bloodstream to transport. I don't remember the name of it. So next are feedback systems. So for our feedback system, we're talking about negative and positive feedback. So these are the types of responses we can get. For negative, the best way I had it memorized is that it always comes up and down. This is how I remembered. And there's like a particular limit that causes it to go back down. Examples of this would be insulin, sugar levels. Basically, it's a cycle of ups and downs where if it's too high, it would result in lowering whatever it needs to be lowered. If it's too low, it will higher. It's in order to maintain homeostasis. I don't know if I explained that yet. Basically, this is just balance within the body. Yeah, so homeostasis. This is the most important factor here. Negative feedbacks are mainly done to keep homeostasis and ensure that everything goes well. Nothing is going too far in one direction. Positive, on the other hand, it just goes up and then it stops. So this is stuff like fruit ripening and the chemical release for childbirth, where it goes up and up until it gets the result it needs, and once it gets its result, it stops. It's also done to maintain homeostasis, but it's more to like ensure everything happens and then just stops. That's the best way I can explain it. I don't really remember anything else within here. I have immune system written here, but there's not really much I can say about the immune system because it was just used as an example. So like, well, not really immune system. Well, protein types. So like, there's T cells, which are used to fight. I believe they're the actual antibodies that fight. There's B cells, which, if I'm not mistaken, this is the signaling where things need to actually go. Yeah, that's really all I can remember. It's just these two. It its responses and uses the bloodstream. So yeah, That's really all I can remember. And that was all for Unit 4, I think. Unit 5 is cell cycle. So I'm gonna first start with like our big circle here. We start with interphase, which consists of G1, S, and G2. This is our M phase, which can be either meiosis or mitosis, but basically prophase, metaphase, anaphase, and telophase. And this is our cytosis, cytokinesis, which can be either cleavage, pharo for animal cells, or cell plate for plant. So yeah, that's basically it. Our G1 phase is just growth. So there's a lot of growing. The cell becomes bigger. We get more organelles. S phase is DNA synthesis and replication. Basically, we're making a duplicate of DNA. G2 is just more growth and making sure everything is correct. And yeah, that's mainly it. I'm gonna now talk a bit about mitosis. So mitosis, it functions for asexual reproduction, repair, replace, and growth. It has PMET. Those are these 4 stages right here. So first in prophase, our nucleus lets go of the nuclear envelope. We start seeing centrioles appearing. Then we go to metaphase where the chromosomes line up and the centrioles release spindle fiber. That's a metaphase. Anaphase, they get pulled to the sides. And then telophase, we get two different. They're not yet separated because that's what cytokinesis is for. But yeah, we rebuild the nucleus, the chromosomes uncondense, and we have a new cell. Things about it: it produces two identical daughter cells. They're diploid, meaning they have the same amount of chromosomes. So, if the original started with 46, then the daughter cells will also have 46 chromosomes. We only have one PMET cycle. So we'll move on from that to checkpoints. So we have major checkpoints. These are checkpoints. They ensured that everything went well. For example, when we were growing the organelles, that everything's okay before the process actually happens. The most important one is this one before G2 because this is before we actually start dividing the nucleus. Oh yeah, M phase is specifically nuclear division. Biokinesis is cytoplasmic division. This is the most important. I'm gonna put a big fat star because it's right before we actually divide. In metaphase, this checkpoint is important because it ensures that everything's actually properly lined up, that not too many chromosomes are going on one cell and too little on the other cell. There's particular reactions by cyclin and cyclin dependent kinase. So, cyclin is technically a negative feedback, to be honest. It goes up and down depending on where we are. So every time it will grow. So like here, it will grow. It continue growing, and it'll continue growing until we reach G2, and then we divide by this. Well, not really divide. We could, but yeah, cyclin continues growing, and then it binds with CDK, creating the cyclin CDK complex. And essentially, they're very important in allowing and not allowing the cell to continue in cell division. While CDK stays constant, cycling does go up and down, which is where the fluctuation happens. More cycling and more bonding, and it allows. There's also, I guess we could put here, G0, which is basically the nothing phase. The cell isn't going through anything. If something goes wrong in one of the checkpoints, there could be things sent to try to repair it, or the cell will send a message, I believe by the complex here, by this complex. It'll send a message to go and commit apoptosis to prevent things from happening. Things like creating tumors or getting cancer. So basically, that's what makes these checkpoints so important. If there was an error here in the metaphase section, we could have people with trisomy. So people who have three chromosomes instead of the typical 2 chromosomes, or a monosomy, which just means they have one chromosome. Obviously, that's Down syndrome, Turner syndrome. We can get mutations as that. We can have people who develop cancer because of constantly growing tumors. From that, we'll move on to meiosis. Oh my gosh, I just remembered something. Yeah, these are somatic. Meiosis goes under some creates somatic cells. In other words, your body cells. So the purpose is sexual reproduction, but its true intention is to have the chromosome numbers. So we go, instead of the normal prophase, metaphase, anaphase, and telophase, we do PMAT twice. These produce gametes, so that's your egg and sperm. So prophase 1, we release the nucleoid envelope, and we can have a process occur. Oh yes, centromeres. Right. We can have a process occur called crossing over. So basically, something like over here, as you can see, these DNA, the genes will transfer over to the other and create unique chromosomes. Yep. Next, we're in metaphase 1, which is it's like normal metaphase, except we're dragging all the chromosomes line up in the center, and we go under the law of independent assortment, which occurs both technically. It occurs in anaphase where they actually separate, but it also occurs in metaphase because this is where they line up in order, while in anaphase 1, this is where the specific chromosomes end up going, which are dragged by the spindle fibers. And then telophase 1, we get our 2 cells. And then we repeat the process all over again. So P2, the 2 cells, membrane disappears, and centrioles appear with phase 2. They line up, spindle fibers. Anaphase, the spindle fibers go in opposite directions. Not the spindle fibers. The chromatids separate centromere. Then telophase, you're getting 4 units. And then we go through cytokinesis to break it all down. So, important things about this, we first are getting genetic variation. So it produces 4 genetically different daughter cells. It also is producing haploid. daughter cells. That means the chromosome number was cut in half. So humans typically have 46. In our gametes, we have 23 chromosomes. This is so when mating occurs, the chromosome number doesn't go over, essentially. We get genetic variation from crossing over and independent assortment. This is how children can look different from their parents. Next, we're moving on to Mendelian genetics. So basically, we got our Punnett squares. Hip hip hooray! So a capital letter equals a dominant. So in normal Mendelian genetics, this is the one that is always expressed. In other words, you'll if this is appears, you are certain that it will always show. So this is like brown eyes that will always appear over blue eyes. This is your dominant allele. Allele is just another way to say gene. This is your recessive allele, and this is the one that is hidden whenever a dominant allele appears. There's different types of ratios. So genotype. So we basically have, well, it's your two alleles, while your phenotype is the trait expressed. The different types of ratios for this one, for example, we have a 1-to-1 ratio. It's just a normal Punnett cross, I guess. Your phenotypic ratio will be a 3-to-1. Then we have our dihybrids. If this compares one type of gene, this does two genes. We know about all of this thanks to Mendel's pea plants, where he did different combinations of peas to study how they would result. So now we're going to talk about non-Mendelian genetics. So these are the different types of when something falls out of Mendelian. Before we actually get there, law of segregation. There is always two types of alleles, and one will always be expressed. Well, dominant allele always expressed. If your dominant allele appears, that's the one that gets expressed. So first we have incomplete dominance. So this is your two alleles not fully expressing. So basically, your dominant allele plus your recessive allele will not have the dominant allele fully go, but it won't also completely remove the recessive allele. So this is how you get your dragon fruit, where you have your red plus your white, and it creates a pink. That's essentially how it works. Codominance, that's what it's called. Codominant, basically both alleles being expressed. So this is where you get your stripes, your rowan. There is multiple alleles, so that you have multiple alleles controlling one thing. So your IA, IBs, and Is. And we finally have polygenetics, where it's multiple alleles controlling one thing. So you like height, eye color, skin color. Those things are controlled by polygenetics. They're not exactly the same because this specifically talks about blood types. But yeah, those are your non-Mendelian genetics. Pedigrees. I love pedigrees. They're kind of fun. So your pedigrees is a type of chart that shows relationships between generations of families. So typically, your circles represent females, your males represent squares. This line represents children. This line represents relationship. There's different types of diseases that can be shown in the pedigree. There's autosomal, which means it's not exclusive to a particular sex. So it could be either male or female, equal amounts. Your recessive will skip generations. So this is like that. While your dominant will appear in all generations, and the person who has it will share it off to their offspring. So sex is sex dominant or recessive. So basically, it's related to your gender. So, for example, for sex dominant, um, all females will eventually will receive this because this is a trait that mainly appears. Females are the only ones that can actually give it. Sex dom regress recessive. Female will be a carrier and eventually will result in a child, typically mostly a guy, receiving it. M- Most of these traits will be passed down by mothers because that's the only way, because mothers contain two X chromosomes while guys could contain one. So the father will always give the Y chromosome to their sons, while the mother will give the X chromosomes. So we got chi squares. Basically, these determine whether or not the genetic ratios are, I guess, expected or out of line. If they are expected, they will not go over the critical value. If they're out of line, that could signify that the recombinant are linked genes. So your linked genes, these are what occurs in crossing over, I believe, or whatever. It's this. They're connected when they separate in your anaphase one. But yeah, that's all for unit 5. So now we're moving on to unit 6, which is all about DNA. So let's start off with the central dogma. So we have DNA that goes to RNA, that goes to protein. This arrow represents transcription. This arrow represents translation. So DNA is double stranded. Ignore how it's spelled. This is single stranded. So this one contains adenine, thymine, guanine, and cytosine. This one contains adenine, uracil, guanine, and cytosine. This one remains in nucleus. This one could go in and out of nucleus. This one contains ribose. This one contains deoxyribose. They both are in a 5 to 3 combination. This is a helix. Moving on, we're going to talk about DNA replication. So DNA is coiled around known as a histone. For DNA replication, we need to uncoil the DNA. We uncoil with a helicase. So this both uncoils and unzips DNA to create our replication bubble. We have topoimerase, which basically makes the DNA stable. Well, not stable, but it prevents the DNA from continuing to break, since DNA is the nitrogenous bases are bonded with hydrogen bonds. So yeah, this kind of prevents it from continuing to open. SSBP, make sure that the bubble is still stable without while it's still detached, right? So we have primase, which was placed down by RNA primase. This is what allows us to place down DNA. DNA polymerase 3. This is what will be producing DNA. What will bring and connect DNA from the primer. DNA polymerase 1 will remove the primer. So number 1 removes the primer. 3 adds DNA. And ligase connects the DNA gaps. Yeah. DNA polymerase 1 both removes the primer and puts new DNA, and then ligase bonds it together to make smooth. Um, this process is semi conservative. So we have both part of the original DNA and part of the new DNA. And that's how you get your DNAs. So this one this group right here, this is a leading strand. That means there is no breaks. While this is a lagging, meaning there is multiple breaks known as Oka something. I'm gonna just put this. It's something with an O. It's Japanese. That means DNA will have multiple breaks that needs multiple ligands place. And yeah, that's your DNA replication. But we'll move on to protein synthesis. So for starters, we'll talk about RNA not processing, RNA creation, I guess, which is very similar to DNA replication, where DNA, well, DNA opens up because of a helicase. We have a promoter or a Tata box, and then other transcription factors over here, over here. So the helicase opens up DNA. We have one that is a template strand. Basically, it allows DNA. It's what codes for RNA. We get our RNA polymerase, and it'll just code until it hits the stop codon. It'll just code the mRNA, pre-mRNA. Yeah, that's basically it. It hits stop, and then it stops making it, and we get our pre mRNA. So from here we go into splicing. So we will splice, which means we're gonna take out the negatives from the positive. So we will only end up with this, with just the negatives that were removed are called introns, while the positives are called exons. So we remain just this. We add a GTP cap to so it can survive exiting the nucleus, and then we add a poly tail so it can survive long enough to actually code the protein. We can make different combinations based off of the same mRNA strand. We can through alternate splicing. So now we have our mature, what's it called, mRNA. So it will travel outside inside the nucleus, and it will go and find a ribosome. After finding a ribosome, it will finding a ribosome. It will be inside the ribosome. We will have tRNA carrying some type of amino acid, and they will enter the ribosome and connect with the codon here. Um, the ribosome will take the amino acids and start creating a chain out of it. Eventually, it will stop. It will stop and will release the mRNA. So it then gets broken down to be reused at a later time. It also releases the amino acid chain, so it could be folded and used at another time as well. I could use like the official terminology for transcription. So transcription, you have initiation. Okay, wait, let me go back to this. Actually, you have initiation, which is actually creating the mRNA strand over here. We got the promoter. We're transcribing, and the mRNA. The mRNA gets created. Elongation is continuing to create the pre-RNA. In termination, there's like a stop codon that releases the mRNA and allows DNA to relax and return to its normal state. Then we go through splicing, blah blah blah, and then we move on to translation. Initiation, the mRNA enters the ribosome, and the ribosome starts reading the mRNA. And then we get the start codon, which is the thionine, or AUG. It's your start codon. From there, we have elongation, which very self-explanatory. We go from one amino acid and we start growing the chain as more and more of the mRNA gets read, with tRNA bringing more and more amino acids. The termination, we reach stop codon. Again, the ribosome gets releases the mRNA. The tRNA returns to float, and the mRNA decomposes to be used at a later time. And then the amino acid chain, the polypeptide, gets released. Okay, moving on. We're going to talk about cell regulation, both in prokaryotes and eukaryotes. There's not really much of a difference. So for prokaryotes, we have our gene. There's two types that gets talked about. We have a inducible and we have a repressible. So we have what we have here. We're gonna call an operator. This is our operator. Over here on this side, we'll have our gene. For an inducible operon, this is what this whole system is called operon. Inducible default is off. So when in a normal state, it's gonna be off. Repressible, the default is on. Oh yeah, cell regulation is just essentially the cell's way of making sure nothing is being too overproduced and nothing is being too. It's to maintain homeostasis, basically. Nothing's being too much produced, nothing is being too little produced. It could be done to like protect, like removing particular features, turning off features in order to survive. So an example of an inducible would be the lac operon, while an example of the repressible would be the tryptophan. So by default, the RNA polymerase will transcribe the mRNA needed from the genes, and then tryptophan gets produced. The more tryptophan gets produced, eventually one's gonna end up in the operator or the promoter. I don't remember what it's called. So once it enters the operator locks, and now our RNA cannot enter and create more. So more of these get removed, and eventually this disappears. And because of it, the RNA can now begin transcription once more. For lac operon, it is always off. For lac operon, so by default, like I said, the operator is off. That means nothing is being allowed. Our RNA cannot go through. So we need something, and for this in particular, we're dealing with lactose, lactose. So the body ingests lactose, and lactose starts entering the body. Some lactose enters, and this causes the whole system to basically move out the way. Now, do I think this is actually what happened? I don't know. I currently forgot how this is supposed to go, so don't blame me for that. Well, do blame me. I'm supposed to. But now our RNA can go and produce lactate, which it uses to destroy lactose and everything. And because there's no more lactose, the operon is off again. That's why this is inducible. You could turn it on to actually do its job. For a eukaryote, the sole difference is that DNA is linear. Well, it's folded a lot. Because DNA is around a histone, DNA has to fold into itself to actually produce, to activate and deactivate stuff. So these are our genes, and this is our activator. There's different terminology, but the process is basically the same. What we do have is a promoter. So the promoter, if you remember, allows the RNA polymerase to actually, one, find area and then to start transcription. So we have a enhancer or an activator, whatever you want to call it. We have our general transcription factors and we have our activator. So when the DNA folds into, so let's say it's this gene in particular, so it folds into here, we get reaction and this will activate our RNA to start transcription for this particular gene. That's the best way I can explain it. Cell determination, I do not remember it that well. Basically, it's the whole it's a process that happens after mitosis. We get our four haploid cells, right? And one of these is an egg cell, and it combines with a sperm cell. They create one zygote. So the zygote keeps growing and growing, and eventually it starts to turn itself. Yeah, I actually do not know what I'm talking about. I don't remember determination differentiation. I do remember something about stem cells. So a stem cell is just a cell that can become anything, specifically for mitosis. So it can become a muscle cell, become a nerve cell, it can become a liver cell, bone marrow, blood. Pretty sure that's what differentiation is. When a cell, after everything, after it goes through, finally has to decide on what role it wants to take. And when determination is essentially the cell, the zygote, because it's probably something related to the zygote, to be honest, having planning the way that it will look. That's Hawk's gene. I remember Hawk's genes, but was that something talked about in differentiation? It's your body plan. So I guess I'll talk about it because I'm not certain if that's what it was. So you have your body. This is your zygote. So over here we have our head, and over here we have our tail. Your Hawk's, and this is determined by Hawk's gene. Your Hawk's genes are your body plan genes. Any changes to the body plan will cause massive changes to how you appear. So if you ever seen people with like fingers that are like stuck together, or just like with multiple fingers, I'm pretty sure that's caused by mutations in the Hawk's gene. The final thing to talk about for Unit 6 is biotech. So we have gel electrophoresis. It's used to determine DNA, RNA, and proteins, like using electricity and a particular type of gel I forget the name of. It finds particular items. I remember in the experiment we did, we used it to determine lineage of things like chicken, mackerel, salmon, tuna. We have plasmid insertion, whatever it's called, related to bacteria, where you incorporate a particular gene into bacteria, and the bacteria goes through goes through protein synthesis to produce whatever the gene was meant to produce. This gene in particular codes, it's used, if I remember the example, it was used for insulin. There is DNA fingerprinting, which helps takes DNA and is often used for stuff like, well, it finds relationships in DNA between things. So it's used in forensics to find who your dad is. There's like a process to do the plasmid thing where you have to take the bacteria and then break the nuclear envelope, incorporate what you want to incorporate, shake it, and then you have to go freeze it, break it, freeze it, so it actually gets in the plasmid. So Unit 7 is, well, the main part of what everybody knows about biology. It's evolution. So this population evolution has three ways of occurring. So one, evolution only occurs in population. One way of evolution to occur is through natural selection. This is where we get our different, what's it called, selection types. So there's directional. This is when a population moves to a particular extreme. So this is like normal beaks grow bigger. We have, I think it's disrupted. This is when a population favors two extremes, as you can see here. So this is like a particular population either going for light fur or dark fur, and not much of the in between, so like gray fur. And then the last one is stabilizing. So this is when a population prefers the middle. Natural selection is not random. It occurs over a long periods of time. It evolves with geno and phenotypes. The point of these adaptations is to both, they're used to survive and reproduce. If the population dies of a particular trait, it can't really reproduce. So the point of natural selection and survival of the fittest is so it can survive, the species can survive and reproduce. So if there's a particular adaptation, that adaptation is done in order to reproduce. Two, we have gene flow. This is, in other words, migration. But essentially, it's having different species interact with each other in order to give each other similar genotypes. Without gene flow, there wouldn't be a lot of biodiversity. Some effects that are caused by gene flow is the bottleneck effect. Basically, in a short period of time, a large part of a population dies. So this is what remains. So we get a decrease in genetic variation. And if you remember, genetic variation is caused by crossing over. Don't start now. Factors to consider for natural selection to occur, there needs to be genetic variation, there needs to be gene flow, there needs to be specific mating. A species needs to only date based off of particular traits. So yeah, moving on, we could talk about the opposite and how we determine a species is evolving, and that's Hardy-Weinberg. This is a model. We have P plus Q equals 1. This is useful for genotype. Well, by genotype, we just really mean the alleles. So P will always equal the dominant allele. Q is always the recessive. P squared plus 2PQ plus Q squared equaling 1. This is for phenotypes. For the word problem you get. So for example, if it's asking for genotype, like how many has this dominant allele, you most likely will use you'll use this one. But if it's asking how much of the population is heterozygous, you'll go use this one. So Hardy-Weinberg is used to find whether or not a population is in equilibrium. This is how we determine whether or not a population is evolving. We know a population is in equilibrium. So, things I do remember is there shouldn't be any gene flow. There is a large population, so a very large population. There must be random mating. So, a population isn't mating based off of traits, little gene variation. For Hardy-Weinberg equilibrium, it's everything opposite. If a population is in equilibrium, none of the things that happen in natural selection is occurring. Next, we have speciation. Basically, as simple as I can explain it, we have population. Population B gets introduced to a new gene. And this population, well, these are the same species, okay? So this population keeps going and going. By this point, right, these two can interbreed. They can interbreed and produce a viable offspring. Species B keeps changing. By this point, we have a bit of a change compared to species A, which is still, remember, these two are still the same species. So by this point, there's been a small change, but these two could still produce viable offspring. So more time goes on. By this point, it's very different from how the other species looks. But they can still reproduce, but their offspring will most likely either won't survive or will not be very viable. By this point, the two species cannot reproduce. They are two different species, and because of this, they cannot reproduce. Yeah, that's basically the concept of speciation, where two species, one changes, they come from the same origin, but one changes and eventually becomes a whole new species from it. So let's talk about reproductive isolation. So we have geographical isolation. I don't remember what the other one is, but it's basically geographical isolation. For example, there's like a giant river separating species 1 from species 2. Because of this big fat river that just keeps going and doesn't allow the two species to go, these two species become very unique to themselves. The biggest example of this is Australia. The other one, I do not remember the word for it, but basically it occurs within the same environment. So let's say we have a forest fire here. This species is going to end up being very different from the species here. So I have here reproductive isolation, but I'm pretty certain that's talking about zygote requirements. So there's not a lot I remember about this. We have pre-zygote and we have post-zygote. So pre-zygote, the requirements for this are stuff like behavior, mechanics, temperature, environment, predators. Basically, before a zygote is formed, everything that goes on can affect the zygote. Post-zygote, that would be like birth, death. So, evidence for evolution. Evidence for evolution will be stuff like fossils. Obviously, I think we all know what a fossil is. I don't think there's any reason for me to go on about those. Okay, so we're talking about homologous structures. There are different types of animals, so humans and whales, but they have similar features. So this would be the fin of a whale and the arm of a human. While they do have different functions, they are structured similarly. Vestigial, which are useless body parts. So, what I was saying is that both homologous and vestigial structures are used to determine if a species has a common ancestor. There's also analogous, structures, which basically are similar body parts, but no real connection between the species. Often, analogous structures are related with convergent, divergent evolution, which basically is two species in different ecosystems that end up being similar, but they're not actually genetically related. So, for example, this species flies, while this species also flies. They're very similar because they live similar lives, but they're not related at all. Moving on, we got cladograms. So there's not much of a difference. This one shows like relationships, I think. When we make a cladogram, right, we have our common ancestor, right? The one they constantly give examples of is great apes. We have a common ancestor, right? And as we go up, they all eventually go to the next. Actually, no. I think this one is related to relationship, and this one is more about traits. This will be those graphs where it's like hairless, species X, jawbone, species Y. I'm pretty certain these are the cladograms, phylogenetic trees, are more in return with the relationships. The other example is these, where they just kind of keep going like this. It's just a different way of showing it. Each point is like a different convergence. We are going to talk about domains and kingdoms. We have our domains, EU bacteria, archaea, eukarya. From here, EU bacteria gets bacteria. Archaea gets archaic bacteria. And eukarya has 4 plants, fungi, animalia, protista. I'll just put green for photosynthesis. Things that can do photosynthesis. We got plantae, we have protists, we have some bacteria. Archaea bacteria does not. They consume with chemiosmosis because they live in relatively high pressure, very chemical areas. Fungi are decomposers. Animalia are heterotrophs. Some bacteria can either be autotrophs, so they can make their own food, so hence the photosynthesis, or heterotrophs. Archaebacteria are heterotrophs. Well, technically they're autotrophs. The last one is earlier. The most specific that I remember is that the first period took forever. There was multiple extinctions. Each of these lines right here is like an extinction. The main thing from this was the experiment where this one dude, I don't remember his name, made the really strange vials. The experiment goes as follows. He would simulate the early Earth atmosphere in these glass vials with the components that you would think would be in early Earth. He would do it in a controlled environment. What was most importantly discovered from the experiment, what was discovered is that you could create amino acids, in other words, something organic, out of inorganic material. Because what was in early Earth's atmosphere, there was no real life. Eventually, cyanobacteria occurred, and we had this large boom of oxygen that changed the atmosphere. But yeah, a lot of that oxygen started appearing. We had a lot of things were changing. Water was now being introduced because we have a lot more oxygen, and more life started coming around. So, in other words, once cyanobacteria started doing photosynthesis, we gained more life. Now we're on Unit 8. And let me tell you, do I remember anything from Unit 8? No, I don't. I will go with what I have, behavior and response, but essentially, actions that species do, so this is the behavior, that respond to stimulus, and it's in order to survive. For example, fight or fight is a type of behavior that is a response to stimulus in a dangerous situation. Ultimately, whatever choice you make is going to be for survival. Energy, the first energy that we ever receive is from the sun. It gives light, and it will go to our autotroph. From the plant, we go into our first consumer. This eats the flower. We got an owl. The owl is eaten by a bear. So each time that we go from these jumps, the amount of energy that gets lost as heat is 10% less. If the producer produces 1,000 energy, rabbit will produce 100. Owl will receive 10, and the bear will receive 1. So there is a food chain, and the food chain is just one type of species X going into species Y going into species Z, going into species A. So this is what a food chain will look like. It's just one of the many types of paths that animals interact with each other and the environment. Food webs, on the other hand, will show the many different species that eat from different connections. While a food chain only shows one example, a food web shows all in a particular environment. Populations and communities. So we have mutualism, commercialism, parasitism, predator and prey. So mutualism, both benefit. This is, for example, crows and wolves. Both have a benefit from the relationship between the two species. Commensalism, basically one benefits while the other one is unaffected. Parasitism, one benefits, the other one is negatively impacted. Predator and prey, one is benefits, the other one dies. These two are negative relationships. This one is positive, this one is neutral. So there's also the equations for population growth, which there's two types. Now, I don't know the equations by heart. I'm not expected to know them by heart, but I do know the graphs for it. So we have our exponential. So this is just population growth that doesn't stop. It just keeps going and going. It doesn't stop. There's no limitation in an environment where there's unlimited resources. On the other hand, we have logarithmic. Okay, this is the one that looks like this. And it doesn't necessarily look like this, but this is the example that's often shown. In reality, it will probably look something more like this, because it's just how populations try to maintain homeostasis. So logarithmic, this dotted line represents carrying capacity. Once it starts getting up there, carrying capacity is the limit that ecosystem can handle of a species. For logarithmic, in this equation, the main important thing is just that you know that there's a carrying capacity, and that impacts how much growth a population will receive compared to exponential. Limiting factors. So, limiting factors. We have both biotic and we have abiotic. So biotic factors would be competition, because you're competing for food, resources, birth rate, I guess, birth, death. Abiotic would be like temperature, pH, erosion. The limiting factors impact your carrying capacity and how much your population will be able to grow. There's particular equations. Do I remember them? No. And I'm not expected to. The next thing is biodiversity. And there is also an equation for this. Do I remember these two? Not at all. But that biodiversity is just how well an ecosystem can adapt. So the thing about biodiversity is higher biodiversity equals more survival, because there is more genes, while lower diversity lessens survival. This is why something like the bottleneck effect that I really didn't get into. The bottleneck effect is when you chop off a good part of the population because of a sudden event. So something like a forest fire, and that kills off a lot of the population. That also lessens the biodiversity because you have less members in your population, which can also expose you quicker to diseases from the outside, because you don't have species that have resistance or immunity to it. Disruptions to ecosystem will be stuff like human impact, natural disasters, competition, invasive species. Invasive species are species that thrive in a particular environment that's not native to it and causes harm to other species. One of the biggest impacted is a keystone species. Your keystone species is a species that, if removed, all layers of the food web are impacted. Basically, your keystone species, you do not want to get rid of them, because if they are in danger, everything else in the ecosystem is going down with it. That is everything I remember from AP Bio. This video is very long, but you know what? There's one thing I can say. I love AP Bio! Thank you for letting me do this recovery assignment. Have a good day.